Good afternoon, everyone. My name is Leandra Fonseca. I am a biomedicine student at the University Unicesumar in Brazil. Before I begin, would you like apologies in advance for any pronunciation errors that might occur during this presentation. Today, I'm here to share with you some results of my research of the mutagenic and genotoxic effects of mercury in the Brazilian human. The present discuss mercury contamination and global issues affects health and environment. If we focus in the last discuss mutagenic and genotoxic effects of mercury on women and of reproductive age and the children. Metallic mercury is used in gold mine and discarded in nature with mine waste. It's transformed into metal mercury, a highlight toxic form that accumulates in the food chain and binds to human tissues, causing health damage. In January 2023, a public health emergency was declared in the Yanomami indigenous land due to poor health condition. Cases include the death of 517 Yanomami children from preventable cases and mercury contamination rates and river exceeding 8,600% of the permissible limits for consumption. This has been extensive report on both Brazilian and international media. The Yanomami, but who are the Yanomami peoples? The Yanomami are an indigenous group living in the Amazonian rainforest region on the border between Brazil and Venezuela. In 2011, the estimated population was 35,000 people, most women and children. They are primarily gatherers and hunters. Some groups have developed subsistence cultivation. Some groups remain isolated with contact with outside peoples. Now, let's talk about the research methodology, the methodology research. The research methodology involved several steps, includes literature research and problem formulation and sets inclusion and exclusion criteria. The aim was to understand the mechanisms of action of the mercury on DNA and these possible genotoxic and mutagenic effects. Data were collected from 8 articles, of with 9 mentions the Yanomami. Exclusion criteria include the articles published before 2908 and the invasion of lands in the region of Minsk was just began. And the articles the present data on urban population involve the manufacturers' activities. The analysis of data considered various aspects. The methodology studies in population is geographic localization, collections and the analysis their main results. This allowed us to obtain the comprehensive and accurate view of the impact of mercury contamination on the Yanomami peoples. War results. Chronic exposure to mercury It can cause changes, movement, visions, problems, shanks, genetic damage, and issues with the immune and blood systems. This is a special risk for developing individuals because methyl mercury can cross barrier in the brain and placenta. Research in rats showed significant changes in the motor cortex and hippocampus, leading to neurochemical dysfunction and behavior change. Increased DNA migration was observation in rats exposed to mercury contamination fish. Expo studied blood cells from Amazonian individuals and found that exposure to low levels of methyl mercury can slow down the cell cycles and cause loss of the cell's ability to multiply. Silderberg suggests that exposure to mercury affects the immune system, leading to right occurrence of immunity disease, infections, and cancer. Cardozo links exposure to mercury, even at low levels, to decrease in the activity of certain white blood cells and a significant drop in CD4 and CD8 lymphocyte levels in humans. Schenker found that T cells, when exposed to mercury, undergo a change in the permeability of the mitochondrial membrane. This is a key event in the cascade activities that led to loss to transmembrane potential in the generation of reactive oxygen species, free radicals, and the league of cytochrome C. Azevedo mentions that the effects of mercury in the immune systems and underestimate and points of the Silderberg divine indicate two types of the observation immunologic effects in the both animals and the humans. The first is autoimmunity, where an immune system produces antibodies against two one proteins and anti-nuclear antibodies from self-reactivity cells, and the increase of IgG and IgA. The authors highlight the reduction of the cellular and the humoral immunity, impartment of the Th1 response, and the increase of the Th2 response. Despite the changes and the effects on the immune system not begin as observation as the damage onto nervous system, animal studies demonstration the induction of autoantibody and production and the development of glomerulonephritis in rats. Conclusion: illegal mine on Yanomami land has led to chronic exposure to right level of mercury. This primarily affects women and children and can cause mutagenic and genotoxic effects in this population. It can lead to change in the nervous system in the both mothers and the children, in addition to change in the immune system. This affects several generations of the Yanomami peoples. For to finish, would like to express my deepest gratitude to my advisor, Prophet Dr. Renata Sanolini, for her invaluable support and guidance during the completion in this work. I also thank the Department of Biomedicine and Pharmacy at Unicesumar. At special thanks to the Science Committee of BSA for the opportunity to conduct this word in the presenting to you. Last but not least, I thank all the participants for their presence and attention, or opportunity to present my work and deep appreciation. Thank you for all.